Hi, I'm Louise Wood and I co-lead the NIHR, and I'm really delighted to be supporting the UK's first Dementia Researcher. Hi, I'm Louise Wood and I co-lead the...
To be successful nowadays in science, you need diversity. You need people from all kinds of walks of life.
We know that for every scientist working in dementia research, there are 4 scientists working in cancer research.
We want to supercharge the next generation of scientists to make the much-needed breakthroughs in both the understanding and intervention in the diseases that cause dementia.
And we have no treatments. We have treatments for cancer, we have treatments for heart disease, but we don't have treatments for the undignified death of dementia.
So nowadays you need chemists, biologists, mathematicians, bioinformaticians, medical doctors, psychologists.
So please think seriously about how you can bring your talents and insights into tackling this awful disease.
Good afternoon, everyone, and thank you for joining us today. I'm Katy Stubbs, and I'm from Alzheimer's Research UK, the UK's largest charity funder of dementia research. And it's my pleasure to host this event today, taking place as part of the Dementia Research Careers Festival. In this event, we will focus on lab-based research, and we'll hear from 4 scientists about what they do, their career path and motivations, and why they think it's a good path for you to follow. So you'll hear about the different fields of study they are in, and also the variety of routes they have each taken. We hope you'll come away with a sense of the growing momentum in the field and how advances in technology, tools and knowledge mean that we're sitting on the verge of breakthroughs. The last decade has seen increased investment in dementia research, particularly here in the UK, and some of the speakers today work in institutes and initiatives established in recent years. This investment has happened because there is a huge clinical need around dementia for more accurate and timely diagnostic methods and effective treatments that can alter the course of the disease. Being a lab-based researcher means you're working at that vital early stage, making discoveries that pave the way towards breakthroughs. So hopefully you'll be inspired by what you hear today and have lots of questions for our panel. What's going to happen is that each of them will talk for about 10 minutes, and then we'll have a Q&A session where we can put your questions to them. You can ask questions at any point during the event using the Q&A if you're watching in Zoom, or the chat if you're in YouTube. And please indicate if you want your question directed to a specific person. So without further ado, it's my pleasure to introduce our first speaker, Dr. Daniel Erskine. Daniel is an Alzheimer's Research UK Fellow based at Newcastle University, where he focuses on dementia with Lewy bodies. So if I can invite Daniel to turn on your camera and your sound, and over to you.
Hi, yes, I'm just going to share my screen. Thank you very much for the introduction, Katy, and to Adam for inviting me to speak today. And hopefully you'll find it a bit interesting, because my path to dementia research has been a bit atypical. So hopefully if you are a bit me and a bit atypical, you will find this particularly relevant. So a little bit about me. I think Katy has kind of summed this up already. I'm an Alzheimer's Research UK Fellow at Newcastle University, and my research focuses on dementia with Lewy bodies. I'll talk a little bit more about that in a minute. I'm obviously not from Newcastle, although I do think I'm getting a little bit of a Geordie twang, but any comments on that, welcome. So yeah, so my research is mostly on Lewy body dementia, and in particular, I'm interested in what causes neurodegeneration in these disorders. So I would say the canonical view in the field is that we get the accumulation of the protein alpha-synuclein into insoluble intracellular deposits called Lewy bodies. You can see them here. And that these induce the dysfunction and death of neurons, and with the loss of neurons of particular functional relevance, thought to underlie clinical symptoms, such as the loss of substantia nigra neurons, leading to reduced dopaminergic input to the striatum, and ultimately clinical Parkinsonism. I don't think it's that straightforward. If you look at things like cell loss, for example, in Lewy body dementia, even though there are Lewy bodies throughout the brain, there's no overt loss of brain mass as a very basic marker of neurodegeneration, and in contrast, the Alzheimer's disease. And the regions that seem to be subject to neuronal loss are those with particularly high energy demands, which is convenient because mitochondrial dysfunction has been implicated in these disorders. And these are similar regions to the brain that you would expect to be affected if you had a primary mitochondrial disorder. So a lot of my research focuses on that. I'm not going to dwell on that too much though. I'm particularly interested in the neocortex, particularly because it contains two types of neurons, pyramidal neurons, shown here in green. These are the only cells of the cortex that manifest Lewy bodies. And in contrast, we have interneurons, which regulate the firing of pyramidal neurons into the rhythmic oscillations that underlie our highest cognitive processing. And in particular, I'm interested in fast spiking interneurons, simply because these have particularly high energy demands. And this, evaluating these two types of neurons is a really nice test of the hypothesis that energy demands of particular populations of neurons may be more relevant to clinical symptomatology than the presence or absence of Lewy bodies, as interneurons do not develop Lewy bodies, but have very high energy demands. And I probe this in a variety of ways. I use microscopy of postmortem human brain tissue. This work so far has shown quite a lot of changes to interneurons, not so much to pyramidal neurons, despite the fact that pyramidal neurons manifest Lewy bodies. And I also use, I think, a very unique system where we culture with organotypic cultures, both rodent and human brain tissue from neurosurgical patients. This is something we're developing at the minute as a unique human model of a uniquely human disease, that being Lewy body dementia. So I wanted to get through that quite quickly to focus on the real subject at hand, which is, where I am, how I got here, and whether this is something that you want to emulate. So, sorry, this isn't working. So my career path has been really atypical. I did not go to university. I did not go to school with the aim of going to university, and I decided to go to university much later in life. So I left school when I was 16. I had no intention of studying anything academic. In fact, if you'd spoke to me when I was 16, or if I spoke to myself when I was 16, it would be I would be really surprised to find out that I ended up in research. I did a variety of jobs, the last of which was working in a bank. It was relatively well paid, but it was extremely unfulfilling. So my job was essentially selling financial products to people, mortgages, guaranteed equity bonds, credit cards, all of these things. And I found it extremely unrewarding. It was quite exciting when I started, but over time it really became very unfulfilling. And this was just before the credit crunch. So this lack of fulfillment made me seek out a more rewarding career. So I decided at about the age of 23, I think, maybe 24, that I was going to go back to go to university. But having not done A-levels, I had to do an access to university course, which was a scheme run by the government in Northern Ireland. I suspect it also occurs in the rest of the UK, which basically is a one-year accelerated program to get you through A-levels to go to university. I went to university at Queen's University, Belfast, and I initially went with the aim of studying the biology of psychiatric disorders, particularly depression. So I come from a part of Northern Ireland which had a very high suicide rate at the time, and I lost several friends to suicide when I was in my early 20s. I was really motivated to do this. But as my final year project was actually developing rodent models of synucleinopathies, and actually I found this a really exciting field, and it was really quite serendipity that I fell into this, but I absolutely fell in love with this subject, and I just became so passionate about it. And that made me seek out postgraduate opportunities that would allow me to further develop this interest. So I then applied to do a master's degree at Newcastle University, as it was quite synonymous with research in Lewy body dementia. So I should say that I was actually offered a master's PhD position at Edinburgh, which was funded by the BBSRC. And I really hummed and hawed about whether I should take this or whether I should go to Newcastle. So at Newcastle, I would have to pay to do a master's degree and then hope that I got on the PhD program. This was a really difficult decision for obvious reasons. But in the end, I decided that I should go with what I love and what I like, which was to study Lewy body dementia. The project that was funded by the BBSRC wasn't on Lewy body dementia. So I did this. It was a bit of a risk. I had to work in a factory all summer to get the money together to go and do my master's degree. So I made pressure gauges and thermometers in Belfast, actually for Ministry of Defence purposes, so it was for tanks. And at the end of that, it was surprisingly well paid. I finally got accepted to do a master's. I worked really hard at it, and thankfully, with my very last application for a PhD, got accepted on the PhD program. So the gamble paid off. And here I am. I've been at Newcastle ever since. Not because I want to stay at Newcastle for any reason, but I think that the research program that we are developing here, and that I'm intimately involved in developing, I think is really exciting. And I think it's giving us increased capacity to answer questions that people have not been able to ask before. And I'm really excited to see where this goes, the foundations we're putting in place currently. So I thought I would also discuss the sort of pros and cons of the approach that I've taken. I'll start with the cons first, so that we finish on a high. So I was a bit older than most of my peers going into the master's program. So I was older pretty much by 4 or 5 years than everyone else, at least 4 or 5 years. And I think there's a constant sense that you are behind everyone because you're that much older. And I think people sometimes expect you to be further on because you are a bit older. So you do definitely feel like you need to do things at a slightly accelerated rate compared to everyone else. But I think there are huge positives as well. And for anyone listening, I think, I really want you to take these on board if this applies to you. I've experienced many different working environments. I have not spent my career in the university system. So I know what life's like outside academia. And actually, it's often very, very difficult as well. You have the advantage in academia that although it can often be very difficult, you are almost certainly doing something that you absolutely love. And it's much more difficult to do a job that's testing if you do not love what you do. And if you love it, and I'm not saying this is a justification of problems with the academic system, but it is certainly much easier to put up with a lot more if you love what you do. I also entered this career knowing exactly what I want. I had a very clear idea in my head of where I am, where I want to go, and what I want to achieve. And I think that this has really helped me because it's not just that I have stayed at university because this is the system that I know. I've come here with a very clear plan that this is what I want to do and this is what I want to devote my life to studying. So I hope that this comes across as some very, I think, great positives. So the question is, should you follow in my footsteps if you are in the same position as I was in maybe 10 years ago, as I'm 35, yeah, so just over 10 years ago. And I don't think it's a simple answer because I don't think research is for everyone. I'm not saying this in some sort of elitist way. It's just simply that it's a very difficult job and it becomes increasingly difficult as you get a family. So I have an 18-month-old daughter, I have a house, and there are unique challenges to research. Working to 9 o'clock at night can make it very difficult for childcare, for example. So it has real world impacts. But I think that my experiences outside research have really equipped me with this. Like I said, I'm doing something that I love, which makes it much more easy to put up with a lot more. It's also the case, I think, that that I have a very, as I said, a very clear plan of where I want to go. So I know that the sacrifices that I'm making are hopefully going towards something that I absolutely love. But I think the most important criterion for research, and I know this is really cliched, but it's certainly something that I feel, having worked outside of research and come into it late, is that having a passion for your field of study and a determination to work at it really are critically important. And I think if you have that, then I think you can almost make anything work. So sort of to conclude, I think it's really important that if you do decide to go into research, maybe later in life like me, that it's fine to take risks, especially if you've weighed it up and the reward equates with the risk that you're maybe going to take. I think it's really important to point out to people that you will find research hard, so make sure you like doing it. And I think that's really important because I knew what the academic system was like. If not before I went to university, certainly during my master's degree, I got a taste of how difficult it can be. But this didn't really put me off, but I was at least informed, and at least I had the desire to work in this field that makes me maybe put up with more than I would have done in a job that I disliked. And I also think, especially if you're a little bit older, it's never too late to start thinking about a career that you may find deeply rewarding. If you have that interest, I think that you shouldn't let it be a barrier. And in fact, as I've tried to point out, there are a lot of transferable skills from working in other fields that I think are very applicable to a career in research. So I will just finish there, and obviously at the end be happy to answer any questions. Let me just work out how I stop sharing. There we are. And yeah, so Katie, should I just mute and turn my video off for now?
Yes, thank you very much, Daniel. Yes, I'll call you back to the screen in a little bit. So thank you for our first speaker there. Just a reminder, you can submit questions at any point and we'll come to them at the end. So don't try and hold them up all until the end, do pop them in now. So on to our second speaker, which is Dr. Christina Toomey, a postdoctoral researcher at University College London. And Christina uses donated brain tissue in her work, exploring the immune system in different types of dementia, with a current focus on Parkinson's. So over to you, Christina.
Thanks, Katie. Yeah, so I work at Queen Square Brain Bank, which is part of the University College London. And I'm a postdoctoral research associate. So I'm going to talk to you today about my career investigating the neuropathology of different types of dementia. So the outline of my talk will go into what do we do at the Brain Bank and how does that relate to my research. I'll then talk about how I got there, so what steps I took. And then lastly, I will talk about my tips and for anybody that would like to follow in my footsteps. Okay, so first of all, what is Queen Square Brain Bank? So as the name suggests, it's a bank of brains, and we collect brains from donors that have movement disorders, dementia, or neurologically healthy controls to compare to as well. So first of all, I thought I'd talk you through a bit what happens to a brain when it comes in to a brain bank, as I always get asked questions about this. So first of all, the whole brain comes to us after it's been dissected, and then we hemidissect it down the middle. So where that red line is in the middle, that's what we would do to make it split into two hemispheres. And the first hemisphere is then dissected into coronal slices, and that's represented by these red lines here. And you get slices that look like the picture on the right. And then these are rapidly frozen at -80 degrees on brass plates. And this stops any ice crystals forming. And this allows us to use the brain tissue for many research applications, especially molecular biology techniques. So to look at the DNA, the RNA, or the protein, for example. The second hemisphere is then fixed in formalin for 3 weeks. So this is like a formaldehyde-based solution. And it basically preserves the tissue so that we can use it for a very long time. The fixed hemisphere is then also coronally sectioned so that you can see by these red lines, we get these slices, and then small blocks are taken from all the key regions that we like to investigate in research or that might be useful in the future, but also the ones that are needed to diagnose the cases with what disease they've had. As at the moment, there are no sure diagnoses in life. We can only be sure when we get the brain at post-mortem. These blocks are then processed, so that means they're dehydrated and preserved before being embedded into paraffin wax. And this preserves them even longer, so we can basically then keep them forever and they won't degrade. We then use a microtome to cut really thin sections of brain. So we've got 8 micrometer sections. So if you think of a millimeter, a micrometer is 1,000th of a millimeter. Yeah, so we've got 8 micrometers, and then these are loaded onto glass slides. And then what we do is a process called immunohistochemistry, which is where we use antibodies to target the protein of interest that we're looking at, and then we can visualize these proteins using this brown chromogen color. So then we can examine all these slides under the microscope, and they can be examined by a neuropathologist firstly to give a diagnosis. And then we can use all of these brains for different research projects. So just to show you an example of this, so for Alzheimer's disease, what we'd first do is look at the macroscopic features, so the ones we can see with our eye. And we can see that in Alzheimer's disease, there's global brain atrophy, and that means shrinkage of the brain. So you can see here, if you compare this normal brain slice to the Alzheimer's disease brain slice, you can see there's an enlarged lateral ventricle, which is the the space where all your CSF fluid lives in your brain. And then you've also got a very shrunken hippocampus, which is where all your memories are formed. We then look at the main proteins that are involved in the disease. So for Alzheimer's disease, this is amyloid beta and tau. And we look under the microscope at the pathological hallmarks that are present in these diseases. And we just assess how many of these pathological hallmarks are present and where they're present in the brain. So for amyloid, the protein amyloid beta, that forms amyloid plaques like this. That's just in the brain tissue. And then it also forms deposits in the blood vessels, and this is called cerebral amyloid angiopathy, or CAA for short. The protein tau also then forms neurofibrillary tangles, and these are inside the neurons themselves. So you can see there's a variety of different pathologies, and this is just the start. So we then look at this for a whole host of different diseases, including all the frontotemporal dementias and the Parkinson type dementias as well. And we can then assess any changes in pathology through the different types of brain. And then I thought I can't talk to you about all of my research now, but I thought I'd just show a bit how we then use pathology to answer our research questions. So one way that we do this is we assess how much pathology there is, as I said, and what shape the cells are that are having this pathology in them. So a way that we do this is by selecting a region of interest on our digital image of our slide that I showed you before, which has had immunohistochemistry applied to it. And then we can select 10 random squares using a Python script, but actually the technique is now advancing even more so that we can do the whole slide at once. And it's becoming very automated. And we're also even looking into artificial intelligence to be able to do this on a much larger scale now. And then we use some software to basically detect any brown that's present in the slide image, and we can then determine the percentage area, the circularity, for example. So these cells here that you can see are microglia, so they're the immune cells in the brain, and they have different shapes depending on what job they're performing at the time. So by assessing what shape they have in the brain, we can then answer the questions about which brains are having different responses. So then to talk about my journey so far. So I thought I'd go right back to the beginning. So I had GCSEs, and obviously my main favourite subject was science. So I did double award science, and that was what first got me interested in science. I then went on to do scientific A-levels for that reason. So I did biology, chemistry and maths. And then that was when I really decided that I wanted to do medicine. I wanted to study medicine at university, and I did apply. But unfortunately, I didn't get in, and it's so competitive that this happens to a lot of people. I've actually written a blog about this, which is on the Dementia Researcher website, so do go have a look at that if you want to know more about this process. But basically, I ended up then going on to study biomedical science at the University of Southampton, actually with Katy Stubbs, that's in this webinar as well. And at the end of my second year, I still thought I wanted to study postgraduate medicine, so I applied again. But actually, when I got to my third year, when I was waiting for those applications to come back, I started doing a lab-based project looking at Prions disease, which is a neurodegenerative disease as well. And that really piqued my interest, and I really enjoyed the work. And I actually then realised that I wanted to go into research. So when I then didn't get into medicine again, I actually wasn't too bothered. I then went to be a research technician because I wanted to gain some more experience before really delving into research to make sure it definitely was what I wanted to do. So I looked at investigating early markers of pathological expression in Parkinson's disease, and we found some really interesting findings about mitochondria with that project. I then went on to do a PhD, and this was looking at the role of TREM2 in neurodegeneration. And TREM2 is a genetic risk factor for Alzheimer's disease. So this is where my interest in dementia really picked up. And this was funded by Alzheimer's Research UK, and was a really good project looking at the inflammation and the microglia like I was just showing you. I then had a little project where I was finishing writing up my thesis as a research technician, and that was working on another type of dementia called PSP. And then I went on to do a postdoc working on lots of different projects between Henrik Zetterberg and Tamron Lashley in the UK and Sweden. And that was a whole host of different dementia type projects before then going on to another postdoc looking at the mitochondrial lipids in Parkinson's disease and Alzheimer's disease. And now I'm just about to start another postdoc before going on to a fellowship. So you can see I've studied lots of different things and done in lots of different positions, so that you don't have to stick in one particular field all the time. So then just to talk about the pros and cons of being a lab-based scientist. So I feel like there's more pros than there are cons, obviously, because I enjoy my work, but of course there are some cons. So I think it's important to be honest about that. So there can sometimes be long days, but the pro to that is that you have very flexible hours. So you can choose when those long days are really. And you can also work it around some of your other commitments, which I find quite useful. There's not always permanent contracts, but hopefully this will get better over time. It's competitive and it can be frustrating when things don't work at first, but that's something that scientists quite enjoy as well, is problem solving. So you can optimize different techniques and get things to work over time. But you can also get to travel very far and wide, normally at least, non-COVID times. The furthest I've travelled for a conference to present my research is Australia, Sydney. So that was a really good conference to go to. I then also have got quite a lot of work published, which is feels quite good to achieve. So I've got 9 different papers at the moment. And it varies from day to day. You can do your independent learning, so you're always learning something new. I love that about this job. You never finished learning. You can always learn something new. And I also like that even though you're not directly helping someone with a disease, you are actually indirectly helping lots of people by helping find the small steps towards that cure. So my top tips for somebody that would like to follow in my footsteps are basically when you get offered opportunities, take them. So this can be anything from big to small opportunities. If you get offered some work experience, if you get offered to go to a talk or to read up or meet a scientist, I think that everything helps when you're trying to get into science. Also get some lab-based work experience. I've also written another blog based on this on Dementia Research website, which highlights some of the organisations that you can use to get some work experience if you are interested in doing that. But you can also contact anyone. Everyone's always happy to answer any inquiries. You can also follow scientists on Twitter. So I find that Twitter is a really great place to keep up with all the latest developments of science and find out what everyone's doing, find out what you like, who you might like to go and work for. I think it's great for that. And then importantly, there's not one path for everyone. So you can see my path wasn't exactly where I first thought I'd go, but actually in the end, it was one I liked. And I didn't do a master's, which is quite unusual for research. So everybody can take their own path and just believe that you will get there to where you need to be in the end. And lastly, believe in yourself and work hard. So obviously science isn't totally easy. So you do have to want to do it. And you do have to work hard, but it's worth it if you really are passionate about it. And as long as you believe in yourself, I think you can get there. So thank you for listening. And if you have any questions at the end, I'd be happy to answer.
Wonderful. Thank you so much, Christina. If you pop off now, then I'll introduce the next speaker and we'll bring you back at the end. Seeing some questions come through, that's excellent. Do keep them coming and we'll have a really good session at the end. So next up is Dr. Ryan West, an Alzheimer's Society Research Fellow and Group Leader at the Sheffield Institute for Translational Neuroscience, based at the University of Sheffield. In his work, Ryan uses fruit flies to unpick the biology of frontotemporal dementia, and he'll tell you a bit about that and his career path. So over to you, Ryan.
Brilliant, thank you. Let me just share my screen. Okay, so yes, thank you for Katie and thanks for the previous 2 talks. So my name is Ryan West and I'm currently a principal investigator and a research fellow at the University of Sheffield, where I work in the Sheffield Institute for Translational Neuroscience, or SITTRAN, which is a designated centre focused on the study of neurodegenerative diseases, bringing together researchers looking at sort of the molecular basic neuroscience end of the spectrum, through to clinical researchers conducting clinical trials and working directly with patients. So in my work in particular, we work using fruit flies or Drosophila melanogaster to understand neurodegenerative diseases, with a particular focus on dementia and a form of dementia, an early onset form of dementia called frontotemporal dementia. So it might seem as slightly odd to use a fruit fly as a model organism to study human diseases. So there are a number of good reasons why we do so. So not only are flies relatively cheap to use and easy to maintain in a lab environment, but flies have a very rapid generation time, going from egg to adult in around 10 to 12 days, and a relatively short lifespan of around 90 days, which means when we want to study a neurodegenerative disease such as dementia, we can do so in a relatively short period of time. So while similarities between yourself and a fly might seem relatively few and far between, we actually know that there's significant conservation of systems between fly and man. So for example, we know that the nervous system has significant conservation, but also all of the genes that we're interested in, or a number of the genes we're interested in, are conserved. And in fact, around 75% of disease-causing genes in humans have an ortholog or a counterpart in fly. But perhaps the most powerful reason for using fruit flies as a model organism is genetics. So we've been using fruit flies since around 1900, when Thomas Hunt Morgan's labs were using fruit flies to understand heritability and explore evolution, Darwinian's theories, and look at genetics. And what they discovered was that heritability was associated with genes, and this was linked to chromosomes. And so because we've been using flies for so long to understand genetics, we've developed perhaps the most advanced genetic toolbox with which we can manipulate flies and understand human diseases. And so flies have become sort of a cornerstone for genetic science in biomedical science, and they're commonly used to understand development, cancer, behavior, but in our research in particular, looking at aging and neurodegeneration. So there's a really good website, the Drosophila Public website, which is run by the University of Manchester, which has a lot more information on using flies in research. So in particular, in my lab, I'm interested in understanding how genes associated with causing neurodegenerative diseases, in particular frontotemporal dementia, cause disease, and ranging from a cellular level through to behaviours. So for example, here we're looking at a fly brain, and we've expressed a gene that we know causes frontotemporal dementia. And when we do so, we see the formation of these toxic protein aggregates within the brain. And so we're interested in how these then affect the nervous system. So we can look at specific individual neurons, and we can see, for example, this neuronal inclusion results in a disruption to the cytoskeleton and organization of these nerve cells. We can then look at a sort of a more functional level and look at how this affects the cell's health. So for example, we know that this axon is essentially the highway of the cell where different proteins are trafficked back and forth. And so we can look at how proteins are then affected by this in their trafficking. So for example, here we're looking at mitochondria traveling back and forth along an axon. So having looked at a cellular level, we're then also interested in how this affects the behavioral phenotypes in the fly, looking at motor circuitry more generally. So on the left here, we have a fly, a healthy fly, that is what we use is an approach called optogenetics. So you can use a light flash to trigger a specific neuronal circuit. So in this case, it's the circuitry that controls the proboscis, the mouth parts extending. And we see that fly has a controlled extension and retraction, nice smooth movement. But when we introduce our mutations that are associated with frontotemporal dementia, we see this results in a neurodegeneration leading to a loss of motor function. So this fly here develops a tremor and it isn't able to control its proboscis in the same way. And so one of the main questions that we're focusing on in my lab, and which is quite a large question in the field, revolves around this gene called C9orf72. So C9orf72 is the most common genetic cause of both frontotemporal dementia and also amyotrophic lateral sclerosis, or ALS, sometimes referred to as motor neuron disease. And so the question we have is how a single mutation, so what's called a hexanucleotide expansion mutation within this gene, can cause two very clinically distinct diseases. So frontotemporal dementia is typically associated with a progressive degeneration that occurs preferentially within the frontal and temporal lobes of the brain, and leads to clinical symptoms, including behavioural changes and language dysfunction. Whereas ALS, the neurodegeneration occurs preferentially in upper and lower motor neurons, resulting in symptoms including muscle weakness. And so using fruit flies, what we're doing is trying to dissect how this mutation contributes to two diseases. And my predominant focus in the lab is on the frontotemporal dementia side, due to my work being affiliated with the Alzheimer's Society. So a little about how we actually got here. So the point of this session is to talk about what we did and how we got here. So you've seen very different approaches from the previous two candidates, but I think my route to where I am now was a bit more straightforward. So I did a degree at the University of York. So I didn't specialise specifically in neuroscience. It was a biology degree with a number of neuroscience modules. But then as part of this, I had the opportunity to undertake a year in industry. So I spent 12 months working for Eli Lilly, who are a pharmaceutical company that specialised in neuroscience. And here I worked as part of a drug hunting team using mouse and rat models of Parkinson's disease. I think this was a real critical point in my career. So this opportunity to spend 12 months working in the lab day in, day out, really emphasised that I wanted to work in research, that I enjoyed it, something that I wanted to pursue. I know some of my colleagues that did the same thing, found this time working in a lab, told them that that's what they didn't want to do, and have moved on to other roles. So knowing that I wanted to continue my research in neuroscience, I went back to the University of York, where I studied a PhD funded by the Alzheimer's Society, looking at frontotemporal dementia. And again, this is where I started my work using fruit flies. After this, I was lucky enough to secure funding from the Wellcome Trust to do what was called a discipline hopping fellowship. So this gave me an opportunity to go away and learn a technique that I'd never done before, something completely different to the field of research I was working in. Again, a great opportunity to learn something new, kind of expand my research horizons, whilst also starting to work on a programme of research that was truly what I wanted to do. I spent a couple of years working as a postdoc, still at the University of York, before I was awarded an Alzheimer's Society fellowship in 2017, which allowed me to move to the University of Manchester and start to establish a real programme of research that was my own devising rather than working under somebody else. Following this, in November 2019, I was recruited to the University of Sheffield at Citran as a PI, which is a principal investigator, where I've had the opportunity to start building my own research group, which is currently composed of my three wonderful students, Joe, Nikki and Duncan, my technician Kate, and this year's MSc student Neve, and supported by funding from Alzheimer's Society ARUK, the MRC and the White Rose Consortium. We are trying to build up a group of researchers focused on using fruit flies to understand the molecular mechanisms underpinning frontotemporal dementia and neurodegeneration. So I think the previous two candidates have given a really good overview of why you may or may not want to do a career in research. And I think I'm not going to cover too many of the negatives. Of course, there are definitely cons of working in research. It's not always an easy career route. There's difficulties in necessarily long-term contracts and job security. You might be moving around a lot, although I think I've shown to some degree that I spent a lot of time in York and you don't necessarily have to move around nowadays. But the real pros are I enjoy what I do. I love working in research. I think I learned this from getting that lab experience from my year in industry. And as some of the others have said, it's a chance to always be learning. We do something new every day. We're looking at something that no one else has looked at often. And so we get to learn new things and do novel research. It's really good to be part of an international community. So you'll work with people from all over the world. You'll collaborate with researchers in other countries. And in doing so, we sort of contribute to a global puzzle. We're all working together on different niche techniques and different pathways associated with the same goal of trying to understand what causes dementia and how, in the long run, we can lead to treatments and preventions for dementia. And then finally, I think something that might get overlooked sometimes is it's a real opportunity to meet inspiring people. So not just other scientists, but working closely with ARUK and Alzheimer's Society, we get to work alongside people living with dementia and the families. And these are the people that really inspire you into why we do what we do and pursuing this career. So as I said, there's time for questions at the end, but you can always contact me. My email address is here, and our lab Twitter handle, which I usually respond to, is West Wings Lab. Okay, thank you.
Wonderful. Thank you very much, Ryan. That was very interesting. Questions are still coming in. That's really good. We've got one more speaker and then we'll move to the question session. So do keep those coming. So next up, our speaker is Dr. Sepe De Shepper, and he is a research fellow at the UK Dementia Research Institute at UCL. And Sepe studies the immune system in the brain and how it changes to drive towards the diseases that cause dementia. So you hear a bit about that and also his career path. So over to you.
Good afternoon, everyone, and thanks, Katie. Thanks, Adam, for giving me this excellent opportunity to present my career path and a bit of the research, what I'm doing at the UK Dementia Research Institute at UCL. So first, I want to talk a bit about my research project. Very simple, very short. Then I want to show you how my career path went until now in science, and also important for those who are chasing a career in science. I would like to give you some tips and some take-home messages, which are very personal to my experiences, of course. So what I'm interested in is how dementia is linked to the immune system, and especially how the immune system goes awry in these diseases. And there is a lot of evidence that the immune system is involved in dementia. For example, a lot of genes that make you, or that make you susceptible to develop some neurodegenerative diseases are mainly expressed in the immune cells in the brain. And also looking at postmortem tissue, so looking at brains of patients, we see that many of the cells of the immune system have been activated. So that means that they are doing things that they normally do not do in a healthy brain. Also, we see in many patients that circulating immune cells really point towards the activation or the involvement of the immune system, what is not happening in a healthy or in a normal, let's say, individual. And so many of these studies, they point towards the involvement of the so-called macrophages, and in the brain, these are called microglia. So the importance of these immune cells in many neurodegenerative diseases. Now, what is a macrophage? As many of you know from your studies of immunology, probably, is that a macrophage was initially characterized as a cell that clears bacteria, that clears any pathogens or viruses that do not belong in your body. And so they clean so that your body becomes free, or that your body gets rid of all the waste and bacteria and pathogens. So initially they were known as the cleaners or the sweepers of the different organs in your body. But it's becoming increasingly clear that these macrophages have many, many more functions compared to, or only in addition to only their sweeping functions. So they be the policemen, they are the doctors, as well as the firemen in almost every organ in the body. And I think the macrophages in the brain, so the microglia, they represent a very nice example of how these cells could have so many different functions. So here you can see in green the macrophage or the microglia surrounding the nerve cells or the neurons in cyan, or in blue Also adjacent to the astrocytes, which are like, let's say, the best friends of the neurons. So neurons need astrocytes to survive. And you can clearly see that these macrophages or microglia closely surround the neurons. And this is important because these microglia, they continuously support and continuously maintain the health of these neurons. Furthermore, if there is any damage or if there is any insult in the brain, these microglia are the first responders to immediately try to clear the situation or try to resolve the damage. And this is exactly what goes wrong in neurodegenerative disease. So many of these microglia fail to do their normal tasks, their normal housekeeping tasks, which will lead eventually to damage or to disease. So for example, these cells, they fail to properly safeguard the nerve cells. They fail to clean the waste or any debris that has been accumulating over the years. And they also fail to monitor and to maintain or to serve the neurons that they surround. Also, what we see is that these macrophages or microglia are so-called overreactive, as we call it, because they are way more reactive than they normally should in a normal brain. And so this is my research focusing about is how these different functions can go awry in neurodegenerative diseases. But it gets more complicated than that because many of these functions that microglia have are not only executed by one type of microglia, but there are many, many different types of microglia. And this is what we call heterogeneity. So different subtypes of microglia exist in different parts of the brain. And so the question that follows is, okay, in neurodegenerative disease, which microglia goes awry and how can we target that particular microglia subpopulation? Now, in addition to my interest in the brain is my interest in the gut. And so the gastrointestinal system has a very similar nervous system and a very similar immune system compared to the brain. And so let's take a look, let's take a zoom into the gut wall. And you can see here in this microscopic image that the gut is composed of a very similar nervous system as in the brain, which we call the enteric nervous system. And these neurons here in red are surrounded by a very similar immune cell type as in the brain, namely the macrophages. And they have a similar function as those microglia in the brain, that is namely to constantly sense the health of these neurons. And if something's wrong, they need to intervene or they need to quickly maintain and to recover or quickly cure, let's say, the neurons. And so the key question is, how do these macrophages respond when the neurons in the gut are at danger? And this is exactly what happens in Parkinson's disease. So Parkinson's disease, which is characterized by typical motor symptoms such as tremor and other disabilities in movements, we see that there is this toxic protein that accumulates in the brain, which is associated with the symptoms, namely alpha-synuclein. But this alpha-synuclein protein does not only accumulate in the brain, but is also found in these nerve cells in the gut. And very interestingly, this is found in many years before the typical symptoms appear in the brain. Moreover, many patients, they suffer from constipation, and which is in many cases the result of nerve cells that are not being maintained properly. And so many of the Parkinson's disease risk genes are also found in the gut macrophages that I just described. And so this is, let's say, one of the other research questions that really drive my research in my postdoctoral scientist, and which has been recently really grateful for my recent funding by the Wellcome Institute for a Sir Henry Wellcome Fellowship. And so this is an example indeed of how these macrophages are really responding to that toxic protein in the gut wall. And so if we zoom in into the macrophage, you can clearly see that the toxic protein alpha-synuclein, here in orange, has been taken up by these macrophages in the gut wall. And the hypothesis here is that once this alpha-synuclein is within the macrophage tends to lose its ability to maintain the neurons and to support the health and the function of the neurons. And so that's the research question that is driving my research. And also, if the cleaning functions are not working properly in these macrophages, how could that potentially lead to further progression of the pathology in the patients with Parkinson's disease, to eventually end up with symptoms related to the motor problems. As this is a presentation for young scientists, I would like to show you my career path starting in 2008, where I started my biomedical science bachelor studies at the KU Leuven in Belgium, as well as my master's studies in 2011. And I think this is one of the things that I would really would tell to every young scientist who has not the best degrees, who has not first-class honors, and which do not have the greatest distinction. So my grades weren't that great. My grades weren't that good, but still I ended up doing what I really love and what I really enjoy to do research, and that is because of persistence, which I will tell you in the next slide. And so eventually my master's degree, I chose the module immunology, and that's where I really got interested in immunology, also thanks to my tutors, which were really passionate about what they were teaching. So that really got me into immunology. A bit later, I started my first laboratory internship, and that's when you really start to work with pipettes and really start to work in the lab. And that's when I realized that science is really something for me. So you don't have to not constantly learn or study all these textbooks that you got in front of you, but you really got to work with your hands, with the background that you acquired. So eventually in 2014, I started my doctoral studies at the KU Leuven, which I graduated in 2018. And I think one of the things that's important to mention for those interested in a research career is that the international conferences are a really great experience to go abroad and to learn other people in the field or other scientists in the field of whom you've been inspired of just reading by their papers. And this is really a way to meet your inspiration in real life and to talk about your research in a passionate way. 2018 was my first author publication, which is of course also a great accomplishment, and which has been of course celebrated properly. In 2019 or in the end of 2018, after my doctoral studies, I decided to go abroad and to get some experience in a lab which is not the home-based lab, which was in my case the KU Leuven in Belgium. And so this is also an advice that I would like to give to all young scientists: go abroad and see how far you can reach. Learn new things, learn new labs, and get to know new people. And so this was really also something that drove my research, is because you get different ideas, you get different inspirations. And then eventually, after my internship abroad, I started the lab of So-Young Hong in the UK Dementia Research Institute, of which I got now, since the beginning of 2021, my own postdoctoral fellowship, which will hopefully drive me towards independency and to get my own lab in a few years from now. And so to end up with some tips from my personal career, and as I already mentioned, is don't be discouraged when you don't have the perfect grades, and it's just the passion about science and the persistence what it matters the most. And so when you start your doctoral studies or any higher education, I would say, find yourself a good mentor who is really willing to take you by the hand, to teach you and to learn new things, and really wants to invest in your career. I think that was for me really important during my doctoral studies. Also have a mental attitude that things will work out in the end. So be positive about the things that you do. And it's not because today the things are not going well or are failing. It can get better, and it will get better eventually if you just don't give up. And you have to have this positive or mental attitude. Also, what I found really important is celebrate all the small and the big accomplishments that you get in uni. So if you have good grades, if you did nice experiments that worked out pretty well, or if you have any papers published. So celebrate with friends, with family, with colleagues. And I think that's really important to remind you what science is about, and that is joy and finding new things, and then moving on after you after you have a positive acquired a positive mindset because of the things that you accomplished. And so that was my presentation. Of course, be curious and be creative. And thanks for listening.
Thank you very much, Sepe. So if I can invite all of our panelists to come back and switch their cameras and audio on, and also introduce Adam Smith, who is going to help us with all the questions. So Adam, do you have the first question lined up?
I do. Let's there's quite a lot of questions, so let's dive straight in. The first question comes from Amelia, who asks to all panelists, how would you suggest getting lab experience in dementia research as a postgraduate before committing to a master's or a postdoc degree? Good question.
I can answer that if you like. So I would suggest trying to do a summer placement project if you're unsure what step you're going to take next, and you want to get some more research experience under your belt. I think there's some really good programs. You can get in touch with different supervisors that you'd like to do that. So I'd probably go for someone a bit more junior. So for example, myself, I had a summer student that was funded by Alzheimer's Society. So they do a summer placement grant that you can apply to. So that's my advice.
Yeah, I mean, I would really mirror that. We've had quite a few students who've come through doing similar things with summer placements, and yeah, quite a few of them have went on to do PhDs in our department, you know, for a variety of reasons, but, you know, just they've got a taste and they really like it because, you know, dementia research is awesome. So of course they will.
Right. Our next question comes from William, who asks, I'm currently in my first year of a biomedical science degree. When it comes to moving into the research field, is a PhD or a master's usually required on top of the undergraduate degree?
Should we do a show of hands for who has a master's before they did their PhD? So put your hand up. Cool. So out of 5 of us here who did a PhD, so yeah, I think it's quite a mixed bag, but does anybody want to take that on as to their views on it? Ryan?
Yeah, so what I would say is when I decided that I wanted to work in research and spent time at Lilly, one of the things that was discussed was that to get further, even in industry, you needed to have a PhD, which you might not have had to have done in the past. And what I'm finding now with recruiting PhD students is there's a lot of competition. A lot of people do now have masters. So it's often the case that you will need a master's or a year in industry to then get a PhD. I don't think you need that to do it, but it's becoming more competitive that more people do have masters, I would say.
Actually, so Christina, you worked as a research technician before you then went on to do your PhD. Are there other people who kind of continued doing that sort of role without ever going to do a PhD?
Yeah, so I think I was going to add to that. Yeah, so I think if you're coming straight off your BSc, you either need to get a job as a research technician and be in it for quite a few years before going for a PhD, or you need to get a master's because it is just that competitive now. And even I've just employed a technician, and even the applications that we got, quite a lot of them had MSCs now. So I think it is still possible if you work in a lab and get that experience. You actually meet people that might want to apply for a PhD for you, so you don't go through the larger application stream. So that's what happened to me. So my supervisor actually said, yeah, I really want you to do a PhD with me. So we actually worked up an application together, and I think that's the avenue in if you don't want to do a master's.
I was just going to add, on our website under careers, under the careers tab at the top, there's an interactive career tool that starts at apprentice level. If you'd like to engage with that tool on the website, you can click and it takes you through the career paths in biomedical research, in academia and in industry as well. And if just having tapped the button to see what that suggests, it says from BSc, the next path would be an MSc, jump straight to PhD, or to go to be a research technician or research officer. And then from that, you can go on to do a part-time. It's usually not research technician to officer to MSc. Usually it's research technician to officer to do a part-time PhD or a full-time PhD. Or you can just stay as, you know, stay on that career path and become a senior research technician and then, you know, run labs and departments without doing the academic side. Shall we take the next question? Timon asks, to all panelists, does your research look at whether psychological factors are a cause for degeneration of the brain, and how does it feel to have the title doctor in your name? We do actually have a specific webinar with some people that might be more relevant to this topic on Thursday particularly. We've got some psychologists and neuropsychologists who are specifically looking at psychological factors and dementia prevention and things. That webinar is on Thursday at 12:30, but if anybody wants to take that question. So just to repeat it again, do any of you look at whether psychological factors are a cause in neurodegeneration, and what's it like to have doctor in your title?
I don't think any of my work.
No, none of you looking at psychological factors.
That
that's the one for Thursday, I think. If you'd like to join us on Thursday, we have a group of psychologists and things specific to.
But it does feel good to have doctor in your name.
Same person asked another question, which is, would any type of lab experience do before you apply for your MRes or PhD, or does the lab have to be specifically related to neuroscience? That's a very good question. Can you go off and work in a different type of lab and then still turn it to neuroscience? Does it matter the experience?
If I can reply to that. So I started my postdoc in UK Dementia Research Institute at the end of 2018. So now almost, no, sorry, it was at the beginning of 2019. So now 2 years. And so before I did not do anything with neuroscience except for studying neurons in the gut. But so no, you can study something completely different. I think going into dementia research is just all about getting familiar with the brain, with any other related neurodegenerative disorders. So it's all about changing fields, and I think in my case, what was my advantage, because I came from the immunological field, is that you get the opportunity to merge your own topic into a new topic. And I think that's where the strength is, of one of the strengths of research these days, is that try to merge different topics. So in my case, that was immunology and neurology to neuroimmunology. I think that could be one of the things that could drive research even further, is first do something else, do like something completely else, completely different, and then start neuroscience with a very different point of view, or a very different, looking at research from a very different angle.
We've got quite a lot of questions, so shall we crack on? Amelia asks, are there job opportunities or areas which are considered stable? As I've heard, it's very uncertain with getting funding of every couple of years, especially with raising families. Is it possible to join a large association such as Alzheimer's Research UK and have a permanent role, or even a team where you get to assist with research? I guess the research technician route, they are longer permanent contracts associated
Not always,
with universities.
but it depends what you're doing.
Well,
yeah. It depends if you're employed on a research project, so that might be short term because it might be only funded for a certain amount of years, or whether you're a general research technician for the whole lab kind of thing. But I think there's also sorry, I forgot what I was going to say now. Yeah, sorry, I'll carry on another time.
But I mean, look, we do also see quite a lot of people who started in science who then apply that in other ways. So that, of course, you can move into industry, or as Katy would testify, I think, Katy, your background in science, and now you have I'm assuming, a permanent job doing science, fantastic science work at a charity. And that goes with the NIHR as well. Many of the people that work with us, their backgrounds are in science. And it's great to have those skills because you can return to it as well in the future.
Yeah, I just remembered now what I was going to say. So there are some institutions that are starting to bring in longer term postdoctoral positions. So the UK DRI, actually Seppe's just gone, but yeah, I think that is a prime example of that. And I think more institutions are hoping to do that, more of that in the future, but it's a work in progress, I think.
Right. Next question comes from Nicole, who asks, thanks to all our speakers for sharing your experience. I'm an MSc clinical neuroscience graduate, and I plan to apply for a PhD research disease pathopsychologically, but I've been struggling to decide what aspects to specialize in since it's such a multifaceted and complex field. I'd really appreciate any advice you'd like to give. So I guess this, I don't know, what do you feel are the hot areas right now? And yeah, go on, Daniel.
Well, not really so much related to Alzheimer's disease, but I think so. There's obviously lots of interesting areas. I mean, I more than most find that I can just get absorbed by the sheer breadth, and there's so many things to be interested in. But I think choosing a PhD is not just about that. Certainly, the subject area is hugely important, but there's other questions as well to be answered, such as how supportive is the PI likely to be? Because these are the things that are really going to help influence your career going forward. So I've been very lucky that I've been, supervised and mentored by some people who've taken a really big interest and really supported me in my career. And that's not just practical things day to day, but also, are they going to encourage you to go to conferences? Are they going to help you apply for funding for this? Are they going to allow you to publish? Now, this sounds really trivial, but, I've seen a lot of people during my PhD, thankfully not in the dementia research field, who would spend a long time studying in their PhD to be mid-author on the paper that came out of it, because their supervisor had to go first and their supervisors, colleagues and collaborators went afterwards. So these things are really important. And I think a really good thing that I certainly thought was worth doing was actually looking into the individual labs you are going to go to and seeing, does this person supervise a lot of students? Are they likely to have time? Do they encourage their students to publish manuscripts, to go to conferences? And I think these are really important questions. So as for the subject that you're interested in, there's so many interesting areas. Inflammation's big, prion-like spread's big. I really like prion-like spread. Mitochondrial dysfunction's big. I like it too. And lots of other people like it, not just the stuff I'm interested in. But I think that it's multifactorial as to what determines a good research program to enter.
So follow your passion and go with the supervisor that seems like they're gonna be the nicest to work for. Next question comes from Selena, who asks, how much teaching is involved in your research careers? Are there many pathways available to academic research that don't involve becoming a lecturer? So how much teaching do you do?
I can answer that if you want. So you don't have to do any teaching in most universities, although it will depend on what university you're at as to how much of research program they've got at that university. But if you're at a research intensive university, you don't have to, so such as UCL. But it is still highly encouraged if you want to go up the career ladder. So for example, I do some lecturing and some journal clubs and things like that, just to start ticking off boxes for me to go up that ladder. So you don't have to. There are people that don't do any, but it's thought of highly.
Okay, we'll take the next question. Amelia asked, now hold on, let me get this right. If you did a BSc in biomedical, but tried to specialize in Lewy bodies within all of your chosen coursework and dissertation, would an MRes or an MSc in neuroscience be better for development? I'd love to be taught more to better my understanding after doing a more broad degree, but I'm aware that the MRes would put me in a good position regarding research. I have lab-based experience as I do work for a pharmaceutical company at the moment, but it isn't in this area. It's quite a complex one, isn't it?
I did an MRes. I thought it was very useful because I got lots of lab experience. And my impression when I was applying for PhDs is that lab experience was something that was valued a great deal by prospective employers. That being said, I see that she already has transferable skills from from the pharmaceutical industry. And as previously mentioned by Sepe, I think that actually having research skills from outside the area you're in is really important, because there's a lot of techniques that we use at the minute in dementia research, and they're the same techniques we've been using for many, many years, which is why innovation is absolutely critical. If we keep doing the same thing over and over again, we're probably not going to advance that much. But actually having new insights from related fields is probably going to be quite helpful. The question is really difficult to answer. I mean, an MRes is really good because you will get experience probably specific to that area. The MSc is good because it will probably give you a lot of taught experience. It depends which you value more and which you think would add more to make you a more attractive candidate.
Fantastic. We have a question on YouTube, which is from Gilberto, who asks, do you ever have bad days in which you just don't want to go into work, or does the passion for science make you want to? Does it sustain you?
Yes. I think that's true of all professions, though, isn't it? So you always have bad days. And yeah, sometimes you can have spent ages on an experiment and it won't work, and that's never a good day. But then another day, you'll get a really good result. So it's definitely highs and lows, I think, in science. You definitely have to be resilient.
Okay, so we've got a question from, I don't know if you're going to be able to answer this one, because I should add this question's from Timon, but I think most of our panel today are the scientists rather than careers advisor peoples. But we do have one of our sessions later on in the week might be more useful, perhaps on Friday afternoon. If a student has a disability or a mental health impacting their performance, would this be considered when offering a place for further studies, and is support available? I think your own institution might be able to offer advice about support. Obviously, I don't know enough about your situation or what stage you're at, whether you're at university right now or still at school. But a disability certainly absolutely should not be a limiting factor to applying to any of the roles in lab-based research. And as for mental health, of course, this is going to be for you to decide, I think, for yourself, if you feel able to apply for these roles. But it shouldn't come up, I don't think, unless you want to disclose it and have that conversation with the supervisor.
I think most universities have a really good support system.
They do. I know, speaking at UCL where I work, they're very helpful and supportive, and that should not be a barrier to you progressing at all, if that's a problem, a difficulty that you have. Selena asks, what are the best things you can do to prepare you for a PhD and a career in research, and to make yourself an attractive job candidate? That's a good question, because obviously now certain universities look for you to have outside experience and things like this. Do you think having perhaps done some, having engaged with people with dementia, having a passion in that way, or does that actually matter in lab research, or is it better to have some hands-on, does pipette skills matter more than passion? Daniel, you were nodding.
I mean, I think absolutely. Meeting people with dementia early on in my career was one of the most pivotal experiences in driving my research. So my grandfather had dementia. He died several years ago. I work a lot with several local dementia groups, such as Action on Dementia Sunderland, who are affiliated with AR UK, but even before I was funded with them, I was affiliated. And meeting people affected by dementia, especially like me and like my colleagues here as well today, I work in a lab with postmortem brain tissue or cells. It's easy sometimes to get lost in the science, and actually meeting people affected by dementia is the most important thing for driving that passion forward. And I always find that I come absolutely reinvigorated by meeting these people to kind of keep working in this area. But in terms of things that you can do during your PhD, aside from public engagement, which I think has had a huge bearing on my career, I think also going to conferences is hugely important. Meeting people, establishing contacts, and aside from anything, it is the only time, let me assure you, it will be the only time that you can go and sit in a bar and talk about dementia research all day without judgment, because you'll be surrounded by like-minded people. And I think this is where fleshing out the science actually gets done, from meeting people, chatting, and establishing collaborations, and that's the future. team science across institutions.
Amelia also asked, what actual work is involved in being a research technician? Which is a very good question.
So it depends on what type of research technician you are. So whether you're, as I say, on one project or on multiple projects. So if you're on one project, you'll be there to really assist with all of the day-to-day activities for that research project to take place. So for example, I was on the one with Parkinson's disease, and I used to get all this brain samples ready. I had my own little side project that I would do. Not everyone will get that, but if you've got supportive supervisors, you might get to do your own little research projects. Yeah, lots of managing little parts of lab life and lots of small little duties really. But yeah, it's very varied. You do lots of different things really.
We have a question from Hannah who asks, hello, I'm an undergrad biomedical student with an interest in neurodegeneration. My question is, how can someone get involved in science communications, especially in terms of dementia research?
Shall I take that one?
I think that would be perfect.
So that kind of science communication and engaging the public with dementia research is the job that I do. And there are lots of different ways to kind of dip your toe in that. It depends how you want to communicate. Is that through written words? So is it blogging, or do you want to work with the media? Is it through kind of events or hands-on activities? So check out lots of different science festivals. There's lots of ways that you can pick up little bits of experience volunteering for some of these different events that are going on. You could have a look at other science blogs and see the kind of thing that they're putting out there. You could guest blog for them or set up your own blog. There's lots of opportunities out there. Yeah, and there's loads of mailing lists as well that you can sign up to find out more like opportunities to get involved with science communication. So yeah, I think we could probably I can put some things in the chat of places that you can look for kind of new and different avenues into it. But it really depends where you want to go with it, but there's lots of opportunity.
And we can probably also as well maybe look to record a podcast on that topic coming up soon. That would be a great one, because we do work with so many people that are amazing in this field. Katy being one of them. Okay, next question. I'm currently in my third year of an interdisciplinary degree. I'm majoring in neuroscience and psychology and minoring in maths and stats. Next year, I'll be doing my master's in the same program, and afterwards, I'd love to do a PhD in something that combines both in the field of dementia. What would you recommend as a good way to get some experience before applying? As I noticed, many PhD applications ask for that. I'll eventually get experience with my master's, but it's too late as applications close early next year. So if somebody wants to get experience in advance of applying for their PhD that won't come from their master's, any suggestions?
Yep, so I have a lot of people that email me to ask if they can come and do some voluntary work in the lab, even if it's a few weeks. Sometimes we can get funding to support people doing this, sometimes we can't, but if you contact a PI or a group leader, most people will happily offer you a chance to get some experience. It just comes down to the person really, whether they have the time to offer it to you and if you can afford to do it. So a lot of these places will be unpaid, which is the problem. But there are, I think Christina mentioned earlier, there are schemes out there to get some funding for summer placements and things that we can do. So.
Well, thank you. Okay, Patricia asks, having studied abroad with a BSc and an MSc, the MSc in Clinical and Cognitive Neuroscience, I moved here 6 months ago in search of research assistant position. I've done 1 year internship in Denmark previously. Unfortunately, so far, there's been no luck. I'm based in London and I am wondering if you have any advice on where best to go to get out there. Maybe not, I did that. I would like to be a part of clinical neurodegeneration research as a research assistant before applying for a PhD. I don't feel I'm quite ready yet. I'm wondering whether this is another question for Thursday, really. unless generally you have any, you know, I think everybody's
Twitter is
suggestion
quite
so
a
far
good
is
way on that instance. Like, that's what I meant by Twitter. I think if you know what you want to go into, follow all the people that you're interested in, and eventually they'll probably post about a position. But otherwise, I would just say persevere. It took me quite a few applications before I got my research tech post.
We do also have blogs on this topic as well on our website, and jobs section as well, which is advertised. And of course, I know the charities often will advertise summer programs and things like that on behalf of individual universities as well. Okay, we're getting through them now. We're down to our last couple. Question here: what are the routes to publishing papers early in your career? I've seen an MSc graduates publishing papers, but how viable is that in terms of difficulty and competitiveness?
It's a very field-specific question, I think. I mean, if you're working with mouse models, it will take a lot longer to publish than if you're working on human postmortem brain tissue, for example, because you know, with human postmortem brain tissue, you have the tissue. With mice, it could be 18 months before you actually have your tissue. So that being said, I think that this sort of comes back to what I said earlier about choosing supportive environments that would allow you to publish papers, and to go for PIs who have a history of encouraging the people who work with them to publish papers. Aside from that, obviously, you know, doing the work, you know, obviously getting through it, reaching a logical conclusion of the work would be the easiest way. But that, as I say, is very field dependent.
Can I add as well? So I just want to point out as well that you don't have to publish at MSc level. Like, I think there's so much pressure on students nowadays because everybody thinks that you have to. I see that coming through quite often. Some people do, but I don't think it's a necessary requirement. As long as you're working and gaining up that lab experience, I think you'll still be competitive.
Fantastic. There's a question from the chat, which I don't think we've addressed, which is it still looked on favourably to have worked abroad, perhaps as a postdoc, or is there now the UK investing so much in dementia research that they prefer you to stay here? Well, of course, as a representative of the NIHR and by that, the Department of Health, I guess we definitely encourage everybody to stay here in the UK and study here. And if you're listening to this and you're somewhere outside of the UK is a brilliant place to study. Please do consider coming and working here. Our website has a whole range of advice and guidance from the British Council, particularly, that can help with the language courses, and all the universities certainly that advertise the courses in our website. Many of those are online now, but they'd also encourage you to consider coming and studying in the UK. But what's your feelings, everybody? Do you have to go abroad now to?
So I would say, I think there's always been an emphasis on go abroad, change labs, even if you're in the UK, but it's not always critical. I spent a lot of my career working in the University of York, and it didn't stop me moving up the ladder. It comes down to each individual. I would say it's very important to pick up new techniques and learn new things, even if you stay in one place, but you don't have to move. Eventually, you'll probably have to move, but you don't have to go abroad. It's about picking up experience. Go and spend a little bit of time in someone else's lab, learn from people within your own institute. Institutes are huge, and you might never meet some of the researchers. You can learn new stuff from them just by going around the corridor. So,
yeah, I've been at UCL my whole research career, but I have gone to visit other labs. So I've gone over to Canada for a month, the University of Ottawa. I've gone to Germany for a short period. I've also gone to other labs in the UK and at UCL. So I think, yeah, you don't have to have a whole position there. I think nowadays that research culture is changing and the message is changing, as long as you are still going on an upward trajectory.
I know we're definitely running out of time now. There were a few outstanding questions which I can take quite quickly myself. Rosa, sorry we didn't get a chance to get to your question, but I've got everybody's email addresses. We're quite happy to send that through to them offline. Jasmine, you asked about undergraduate medic. We actually have a webinar on Friday at 12:30, which I hope you can join, which is hosted by the NIHR Academy, who can talk about the doctoral fellowships that you can do as a clinician, and how you can become involved as a clinical academic. And we also have some people who work in the NHS at that session on Friday. But if you have a question about how to not use your medical degree and go straight into the science, by all means, drop me a line in the chat and we can follow up on that. And as for Ashes, who asked a question on YouTube, followed research in Australia in dementia, would organisations in UK take international applicants? Of course, absolutely they would. There are jobs advertised on our website and in the learning section of our careers website, and some links and resources that you'll find useful there as well. I think that's all for the questions then, Katie. Thank you.
Wonderful. Well, thank you very much for handling all the questions, Adam. We got through quite a lot, and thank you everyone for submitting them. And a huge thank you also to our panelists for sharing your research and also the huge passion that you have for it. That's been really, really interesting to hear. This event has been recorded, so people can watch this back, or you can share it with others who weren't able to attend today. And just to flag, there are other webinars happening this week. So we mentioned a few of these. Some of them are on things like data and technology, looking at psychological research or research using arts and culture, and lots more. So check those out on the website and sign up for some of the others if they interest you. There's also going to be a social event at 4 p.m. on Friday, so hopefully some of our panelists today will be able to join us for that. And it's a chance for you to come along and interact with them and ask them some more questions if you'd like. So check out the details for that also on the website. Thank you once again to our panel. It's been absolutely fabulous. Thank you for sharing your time and your insight and your experience. It's hugely motivating, and hopefully everyone listening has been inspired by what they've heard and kind of the different puzzle pieces that you're each adding to our overarching knowledge of dementia and how that's putting us on the path to breakthroughs. So thank you so much to panelists. Thank you to attendees for coming. I hope we've inspired you, and have a great day, and hope you can join us for more events in the series. Thank you very much, and goodbye.
Thank you. Bye.